Can anyone see my slides?
Yes.
See
Yeah.
your
So is it
X-ray.
now in the full screen mode?
Mm.
Okay.
Can you scroll up and down, see whether it can move well?
Is it moving? Hold on.
Not moving here. Yep, moving.
Okay, so it's moving already, right? Okay. So can I start now?
Yeah, sure.
Oh, so okay. Then I'll start now. Do you need me to switch on my camera?
This is your first slide.
Yes, this is my first slide.
Where's the title?
Where's the title?
It's okay, it's okay. Later you'll see the title.
Maybe you can turn on the camera for a while.
Yeah, okay. Let me just. Can you see me?
So your supervisor is not here, yeah? Okay, is that okay to you?
Yes, it's okay because he told me that he's outstation. Yeah.
Right. Okay, fine. Maybe I want to see your title.
Oh, because it's the third slide on my title. My title is
Okay, so just go ahead. Okay.
So I'll go ahead, yeah.
Yeah.
Okay, so good morning everyone and good morning to panel of judges. Do you know that TB remains as the major public health threat in which it kills 1.6 million of people per year, placing it the second leading cause of death from single infectious agent? So today, the topic of my study is somewhat related to TB, in which the title is Expression and Purification of TCR-like Antibodies Against Latent TB in Bacterial System. I'm Ding Yi En, and under supervision by Associate Professor Dr. Tai. So TB is an airborne infectious disease caused by Mycobacterium tuberculosis. The clinical signs and symptoms of TB are as shown on the slides, which includes blood cough, fever, chest pain, chills, and etc. And is TB deadly? The answer is yes. Is there a cure for TB? The answer is also yes, as we have antibiotic regimens. However, is TB eradicated? The answer is unfortunately no. One of the major contributions for why TB is really hard to eradicate because there are different stages of TB infection. So during primary infection, 70% of the individuals are actually able to eradicate this bacteria by their own host immune response. However, 30% of these individuals will get infected, and in which 10% of these individuals will progress to an active TB infections, ADB. And these individuals will then manifest disease signs and symptoms and also transmit the disease to another person. Majority of the individuals, however, which are 90%, will progress to a stage known as latent TB infections, LTBI. So latent TB infections is defined by a persistent immune response towards MTB antigens without evidence of TB symptoms. Globally, it is estimated that 1/4 of the populations are actually infected with LTB. Additionally, these individuals are having 5 to 10% lifetime risk to reactivate to an active disease. This highlights that LTBI is a future major reservoir for active disease cases. Looking at how LTBI individuals do not manifest disease signs and symptoms and also do not transmit the disease to another person, it is clear that LTBI individuals is really hard to detect in the social settings as all of us look normal. Unfortunately, currently we do not have a gold standard for LTBI diagnostics. However, we do have commonly used diagnostic tools in the clinical settings, which include TFT, tuberculin skin test, and IGRA, interferon gamma release assay. The principles of both tests are based on the detection of immunological memory of our immune systems against the MTB antigens, both in vivo or in vitro. However, this suggests a major limitations or significant limitations of current LTBI diagnostics, as it do not provide a direct measurement of the antigen itself. And apart from that, current LTBI diagnostics are unable to differentiate between ATB, active disease, and a latent TB infection. So looking at how TB is a major public health threat, and there is a need for a new diagnostic alternative to tackle these limitations, and one of these possible entity is TCR-like antibodies. Before we dwell deep into what is TCR-like antibodies, let's just have a general overview of the adaptive immune system. So our adaptive immune system are generally categorized into two arms, which are the cellular-mediated immune response and humoral immune response. For cellular-mediated immune response, the TCR recognition on the pMHC molecules, which are the peptide bound to MHC molecules, on the cell surface are very important for T cell activations to elicit their effective functions such as T cell proliferation, cytokine secretion, and cytotoxicity. On the other hand, for humoral immune response, it is responsible to produce antibodies which will tag on extracellular antigens to mediate their effective functions, which includes ADCC, ADCP, CDC, and etc. So what if I tell you that there is one entity that could bridge the mechanisms of both cellular-mediated immune response and the humoral immune response? And this entity is TCR-like antibodies, which were shown to be able to recognize pMHC molecules and also to mediate the effective functions of an antibodies. The mechanism of actions of TCR-like antibodies is not just limited to the ones that's shown on the slides. However, the mechanisms of TCR-like antibodies are actually majorly subcategorized into two categories, which are antibody-dependent and T-cell dependent. For antibody-dependent mechanisms of action, it relies heavily on the Fc portion of the TCR-like antibodies to elicit their effective functions as shown in previous slides, which includes CDC, ADCC, ADCP, and direct apoptosis. On the other hand, these antibodies can also be conjugated to a drug or toxin to deliver toxic payloads onto the target cells, thereby killing them. On the other hand, in T-cell dependent mechanisms of actions, TCR-like antibodies act as a linker to redirect T cells to recognize and target the infected cells. Thereby, it is able to elicit their effective functions to kill the target cells. So collectively, many studies has already suggested that TCR-like antibodies is a promising tool for therapeutic applications, especially in cancer and infectious disease therapies. And this is attributable to the ability of TCR-like antibodies to tag or to target intracellular antigens that are presented on the MHC molecules. So now the question is, why TCR-like antibodies, and how is TCR-like antibodies suitable for LTBI diagnostic and therapeutics applications? So first of all, Mycobacterium tuberculosis is an intracellular bacteria, as it resides within hosts for persistent survival. And this suggests that TCR-like antibodies is suitable for this MTB diagnostics, as it is able to tag on intracellular antigens that are presented on the MHC molecules. Apart from that, TCR-like antibodies has strong binding affinity. As according to studies, TCR-like antibodies has 10 to the power of 3 to 10 to the power of 5 stronger binding affinity as compared to that of the TCR. As a strong binding affinity is a foundation for a good immunoassay development, this suggests that TCR-like antibodies can also be used as an immunoassay in the future. Apart from that, as shown in previous slides, TCR-like antibodies can elicit different mechanisms of actions. This suggests that TCR-like antibodies could be also used for LTBI therapeutics, as it could be a potential complementary drug delivery system and also mediate the Fc effective functions of an antibodies. This is especially true when we are at the end of the road of antibiotic treatments. And last but not least, just like the monoclonal antibody, TCR-like antibodies has the practicality to produce as an off-the-shelf therapeutics as compared to other immunotherapies that are cell-based. Collectively, this evidence suggests that TCR-like antibodies are suitable for LTBI diagnostic and therapeutics applications. So since TCR-like antibodies is so promising, my previous seniors has already generated these TCR-like antibodies to tag against antigen from Mycobacterium tuberculosis. And this antigen is the 16 kilodalton heat shock protein, which was primarily produced during MTB dormancy. Many such studies has also suggested that detecting this antigen could have the potential to differentiate LTBI from ATB, suggesting the diagnostic applicability of this antigen for detection of LTBI. Apart from that, they have also developed generated HLA molecules that could able to tag on binds to this 16 kilodalton heat shock protein. And later on, they have generated TCR-like antibodies through phage display technologies and show the ability of these TCR-like antibodies to tag on their kinetic antigen. So the challenge of this study lies in how to bridge the discovery part of the TCR-like antibodies to the potential diagnostic applications of these TCR-like antibodies in the near future. Hence, there is a need to express these TCR-like antibodies stably in both rapid and economical way. So to achieve this, we are choosing bacterial expression system, as many studies suggested bacterial expression system has several advantages, including cost effectiveness, easy manipulation, and fast replication rate. However, bacterial expression system can come with several disadvantages, which includes minimal post-translational modification and inclusion bodies formation. Luckily, studies has also revealed that by manipulating culture conditions, including induction temperature, induction time, IPTG concentration, and etc., they can reduce the inclusion body formation. Hence, this leads to the objective of my study, which is to optimize HLA-A11 TCR-like single-domain antibody expression in E. coli expression system. The hypothesis of the study includes optimizing the growth and induction protocol can significantly increase the yield of TCR-like single-domain antibodies, and these antibodies can be produced in the functionality of these antibodies can is less affected by this expression. The specific objective of these studies are as shown on the slides. Okay, so first of all, I have to clone this TCR-like single-domain antibodies gene into a suitable vector. Secondly, I have to optimize the bacterial expression condition for this protein expression. And lastly, I have to preliminary confirm the functionality of these antibodies against their antigen. To achieve my first objective, which is to clone the target genes in the suitable vector, the general overview are as shown on the slides. So briefly, I have to isolate the plasmid with target genes and also select a suitable vector, and perform a double restriction enzyme digestion for both of the vectors, and perform a gel electrophoresis to separate the DNA bands. Once DNA bands were separated, I will isolate the target genes through gel extractions and ligate the DNAs together, and transform this ligation mixture into a bacteria. During the second day, I have to select a positive clones and send perform a plasmid extraction to send the DNA for sequencing. And last but not least, the confirmed DNA sequence plasmids will be transformed into an E. coli for protein expression. So for protein expression, I'll have to culture E. coli containing the target genes overnight and dilute it until it reaches 0.6 OD. And later on, I have to optimize the induction conditions, which includes temperature, IPTG concentration, and incubation time. Once optimized, I'll have to extract the extract the proteins through periplasmic extractions and purify these proteins through immobilized metal affinity chromatography. Last but not least, I have to determine the protein concentration through nanodrop and determine the protein identity through STS page and Western blot analysis. To achieve my objective 3, which is to preliminary confirm the functionality of these single-domain antibodies that I've expressed, I have to perform a dot blot. So briefly, I have to dot the single-domain antibodies onto a nitrocellulose membrane and let dry. Later on, I have to block the membrane with 5% BSA and wash 3 times with TPST. Next, I have to incubate the membrane with biotinylated 16 kilodalton HLA11, which is the connect antigen for my single domain antibodies that I express. And further, I will incubate with HRP conjugated streptavidin for a detection. Last but not least, I will stain with diaminobenzidine for detection and result analysis. So for expected results, we are expecting that I am able to clone these target genes from a pcDNA 3.1 into a bacterial expression vector. Upon that, the bacterial expression will be successfully optimized for production of these HLA-A11 TCR-like single-domain antibodies. And last but not least, these antibodies that are produced are all functional. The research activities are as shown on the slides, which I will start on October of 2024 and ends as we are expecting to end these research activities by September of 2025. Marked with asterisk signs and M is the milestone of my studies. M1 is to achieve cloning of target genes from pcDNA to PET vector, and M2 and M3 are shown on the slide. Yeah, so these are the references, and thank you for your time. That's all.
Okay, thank you. Ki En,
Yeah.
Ki En, okay. So, shall we invite our evaluator for the questions? Any questions from the evaluator? Can we start from Dr. Venu?
Hi, good morning. Thank you, Dr. Koo. Hi, Ian. Good morning.
Yeah, good
How are
morning,
you?
doctor. I'm good.
Okay. So, nice, very good presentation. You presented very confidently, and the way that you presented is good. So, can you go back to your problem statement slide?
Problem statement. This one.
Yes. Okay. So, what is the current problem that you are having? Because you want to develop some kind of potential diagnostic, point of care diagnostic. So what is the main problem with your current diagnostic that you're not getting what you're supposed to get?
Okay, actually we are not progressing until the diagnostic part yet. Currently we are just trying to optimize the expression of these TCR-like antibodies so that they can be produced in a large scale for testing in the later stage for future application for diagnostics.
Yes.
Yeah.
So your main idea is to produce it for future diagnostics, future
Yes.
TB diagnostics.
Yes, correct.
So there should be a reason that you want to produce this for future diagnostics because there's a problem with current diagnostic.
Yeah, okay. So the problem of current diagnostic, we have to go back to limitations of current LTBI diagnostic tools, which are shown in this slide. Current diagnostic tools do not differentiate between active TB and LTBI, and also it provides an indirect measurement of immune response against MTB antigens. That's why we are trying to develop a single domain antibodies, which are TCR-like antibodies, which might have or a promising ability to differentiate both ATB and LTBI, and also provide a direct measurement of these MTB antigens instead of providing a immune response based measurement. Yeah.
Okay. So because when you explain, you said might, okay, your single domain antibody might be able to differentiate between ATB and LTBI. Okay. So you're proposing something that might be, or you confirm that your TCR-like antibody could differentiate between ATB and LTBI.
Okay, theoretically it should be able to differentiate between ATB and LTBI. However, because we haven't tested it on the, I'm not sure if they tested or not, but currently from my understanding, they haven't tested it on the real patient with ATB and LTBI. So I cannot be confirmed 100% saying that this is able to differentiate and or not. Yeah, that's one of the concern. But we are trying to continue, further this investigation. Later on in the biosensor development, perhaps we have biosensor development, and in that case we can try on to differentiate, try on the patient sample to see if this diagnostic tool can really differentiate ATB and LTBI or not. So now I think we are at the very initial part of this study.
Yeah, I do. Okay, you see, why I ask you this is because when you presented, I couldn't see the important, because whenever there's a research, okay, the problem statement is very important. Okay,
Okay.
why you are doing this work, why you want to do this work, and what you are trying to solve. Okay,
Okay.
there's a gap there that you didn't explain it. Okay. So,
Okay.
like even now that you are saying that it might happen. So when it might happen, then why you want to do it before you have some kind of informations or data, then the whole experiment goes out like that. Okay. So there should be an statement saying that, okay, TCR-like antibody has the ability to differentiate between ATBI, okay, and LTB because of A, B, C, D, E. So that is why that we are starting to produce this, which in future could be used for diagnostics. So even in your thesis, that kind of statement must be there. If not, then you have to change your whole problem statement. Or you cannot put POC, you have to put something else for you to come up. And do you get what I'm trying to mean here?
Yeah, but actually
Yeah.
in this study, I already written that this antigen is the potential antigen of the study. That's why we are doing this.
No,
I'm not
yeah.
sure is this correct or not.
No. I think I think you are not sure, but your boss, your Dr. Tai is sure why he's doing this, and he might have the information. What I'm trying to say, you need to include that information in your problem statement for you to
Okay.
be more
Can
clear. I'm not saying that you need to change your antibody. No, I'm not saying that.
okay.
Okay, I'm saying that you need to add in informations why you are doing this.
Okay,
Okay,
understand?
why? So what is the connection? Okay. And then as the same thing that you are using HLA A2, A11, and A24. So what is the reason there's 3 HLA has been chosen?
Previously it was found that HLA-A02 antigen or antibodies are antigens are commonly seen on the world population, and HLA-A11 and A24, including HLA-A02, is prominent in Malaysian population, 70% of the Malaysian population. So we are targeting, if we are to produce this biosensor in the future, we are targeting actually Malaysian population for this usage. Yeah, that's why we are choosing this HLA A02, 11, and 24. But currently in my study, I'm only producing the single domain antibodies for HLA A11 due to time constraints. Yeah.
Okay, I got it. So because your time is short, you're just taking in A11. So why specifically you choose A11 rather than A2 or A24, or A2 and A24 somebody have done it?
Actually, there's no reason. I just choose one because yeah lor, I really just choose one to be honest lah, because due to time constraint. But if I have the time, I would try to do all of this.
Yeah, I think that one also you must tell, because there's a reason, and there's a population, as you said. HLA A2 is about 40% of the world population. HLA A2, A11, and A24 is about 70% of Malaysian population. So that kind of statistics also you need to give while you are presenting, to tell that why this thing is being chose.
Okay, can.
Okay. So can you go to your methodology slide?
Methodology, this one.
Yeah. So this is your cloning part, right? Okay. So where's your So are you going to do any biopanning? I could, sorry,
No,
I didn't get.
actually the biopanning has already been done by my senior part. They have already developed this TCR like single domain antibodies. I'm just cloning it into a suitable bacterial expression vector because previously they have done on mammalian cells instead.
So they have done all the selections,
Yes,
clones,
yes, yes.
positive clone
I'm
selections.
just trying to express this protein instead in a mammalian vector, in a bacterial vector instead because it is more rapid and it is also more in an economical way.
So I think while you're presenting, you should have just tell us the earlier part. This thing has been done because whenever you talk production and expression of TCR-like antibody, there's a very big cycle of peptide selection. Then
Yeah.
you are doing biopanning and selection of clones and things. So while I look at your methodology, you straight away I could understand that you have done it. Somebody have done it for you, and you're just taking the clones and using it. So now you have to explain that is much more better. And then because
Okay.
people have done it in what I'm suggesting, people have done it in mammalian cells, and you are now trying to differentiate between the mammalian cells and also bacterial expression. Okay, trying to improve the expression in bacterial expression something, I'm not sure. So
Correct,
that is
correct.
the idea. So it could be the way that you are comparing. People have done it earlier one, then you are comparing it. Okay.
Actually, to be honest, I'm not comparing to the mammalian expression. I'm just trying to change the mammalian expression to a bacterial expression because it is more economical way and rapid way to do it. Yeah.
Okay. Okay.
Okay.
So so your next slide, please.
Next slide.
This is your purification. So you are choosing ISTAC. You are choosing ISTAC because that it is all the histidine tag is already in that, or
Yes. Because the histidine tag is already inside of this expression vector. Can you see my slides? We are choosing actually PET51B expression vector for this study because this plasmid actually contains of the 10 times his tag, so that we can actually purify them through immobilized metal affinity chromatography in such case. Yeah.
Okay. So may I know that have you started to do any lab until now?
Actually, yes.
Any work? Okay, so you are at which stage?
I'm at second objective already,
So
which
you
is
have
the expression.
So you
I've
have
completed the
your
first
expression.
objective.
So now you have completed the first objective, which is
Yes.
your cloning is
Cloning part.
done.
Yeah.
So go back to your previous slide, your objective one.
Okay.
So how you verify that your cloning is done? By sequencing?
Yes, by DNA sequencing. So previously I've already sent it for DNA sequencing, and yeah, the results come out okay. How to say it? I checked the sequencing result.
Okay. Okay. Good that you have done your first objective, and you have one more objective to go.
Doctor, time
And,
constraint, doctor.
yes, we understand that for mixed mode. Don't worry, we are not asking you to go and do big things. Okay.
Can.
So you have only 2 objective or 3 objective? Sorry,
I
just
have
now
3. So the last
Yeah.
objective, I have to confirm if this antibody is functioning or not. Is this antibody is able to bind to their connect antigen or not? Yeah.
So you will be how you will do this objective 3?
I will perform dot blot, which
So
is
you're going to
the
perform dot blot using the positive antigens.
Yes, the antigens are as shown here. This antigens is the peptide bound to MHC molecules. So once
okay.
if this binds, means this TCR-like antibody can function.
Mhm.
Mm-mm.
So what is your plan B if they don't bind?
If they don't bind, okay, by right it should bind lah. I haven't
Yes,
go to
by
that
right.
stage that if it doesn't bind, because if it doesn't bind, the whole part of the study would be kind of pointless to me. But it should bind lah. But
No,
I'm
no.
not sure what is the plan B
Theoretically,
for
no,
this
that's why I'm
yet.
asking. Theoretically, it
Mm.
should bind.
Mm, mm,
So
mm.
when you have already expressed it, purify it.
Mm-hmm.
When you're trying to bind it, if it doesn't bind, so what is your idea and what is your plan? So you should think about that and
Okay,
how you're
can.
going to troubleshoot where the error might have gone. Okay, it could happen. It could happen.
Yeah,
So
I can understand that.
possibility is there.
I
Yeah.
can understand that unforeseen circumstances can happen in lab. I can
Yeah.
go and discuss this with Dr. Tai and see what is the plan B about this, and also go
Okay.
and find out. Yeah.
Okay. Thank you. Thank you.
Okay, thank you, Dr.
Thank you,
Tai, Dr. Vanu.
That's all from me, Dr. Koo. Thank you.
Okay. Dr. Liao, right? Dr. Liao, do you have any questions?
Okay, thank you, Dr. Koh, and well done for Ian for your great presentation. I'm sorry that I lost my voice today, and you're so lucky, so I don't have too many things to ask, but I still have something like to chat with you.
Okay.
And thank you for Dr. Vanu for raising up so many good questions to you. I hope that you can jot down all of this. So for my part is you mean that you want to differentiate about the AT and LTD. What kind of antigen are you looking at?
Oh, we are looking at this antigen. Wait, yeah, this. Can you see my slides? Okay, so
Yep.
this is the antigen that we are looking at. It's the 16 kilodalton heat shock protein.
Okay, 16 kilodalton. Is it 16 kilodalton heat shock protein only present in, like, for example, ATD or LTD?
It's only present in LTBI individuals, not ATB, because according to studies, this heat shock protein is primarily regulated by this DOS-R regulon of the Mycobacterium tuberculosis bacteria. And during granuloma formation, which is during the LTBI, they will, because inside of this granuloma is very acidic and there's hypoxic and all that, this antibody, this bacteria will produce, which will activate this DOS-R regulon to produce this to produce this protein for their survival. So it is majorly produced only during MTB dormancy.
All right. So is that this antigen 16 kilodalton that presented internally or externally?
What do you mean by internally
Internal
or
cells in the intracellular or extracellular?
It's an intracellular antigen presented on the MHC molecules. So I guess it's externally presented to the TCR lah, or else it wouldn't bind.
Okay, and because that one is when you do it, when you apply for the therapeutics is good, because diagnostic sometimes is a little bit different. Okay, as you say that this is presented internally, and you want to turn this so-called the TCR antibody to be the diagnostic agent, and do you need to make sure that this protein can be presented in your sample externally?
There, I think from previous study they have already tried on this, and it's not like they tried, they have searched on the database and show that this HSPX protein is actually presented on the extracellular. It means it is presented on the MHC molecules.
So okay, other than that, have you because you're just starting, you're not doing anything and starting anything like for the budding yet, but have you checked the literature review saying that is this 16 kilodalton protein only can only be found in this? It's a unique, I would say it's a unique protein that found in this tuberculosis. And okay, my question I'd like to check with you is like, how long is the amino acid of this 16 kilodalton protein can be detected by the TCR antibodies done by your
Um,
lab previously?
Previously, I think for HLA11 is around 9 moles of amino acid bound to this PC. PMHC was detected only these 9 moles.
9 months. Okay, thank you so much. Okay, yeah, now I go to the next. Okay, in your study, because you are not going to use the mammalian expression system to produce this TCR like the big antibodies. However, you are focused on the single domain antibody. Try to determine what is the single domain antibody that you define in your study?
I think it's the VH region of the antibody of the domain.
VH region. Okay, good.
Yeah.
Why do you want to use only VH, not VL?
Because VH and VL together, it will become a big protein, and it if expressed in bacterial expression system, I don't know. I don't know if we express SCFV the whole thing, it would be problematic or not in bacterial expression system. But I actually saw many studies has also produced this the whole thing in bacterial expression system. Currently, VH has increased solubility and increased stability as compared to SCFV. That's why we are producing a VH. Yeah.
Okay, now as you mentioned that the VH has already been increased the solubility and stability. Can you explain it? How do you improve the solubility and the stability?
Of the VH.
Yes.
I don't think I can answer your question at this stage because I have quite limited understanding as compared to you on the antibody production. But I can always go and check and come back to you after this.
No. Thank you so much. I'm not asking for any you compare whatever because the hypothesis is quite important. As you mentioned that you want to produce a single domain only VH only antibodies. The stability is quite important. And Ian also mentioned that your antibody is already quite stable in terms of blah blah blah. Because following the question asked by Dr. Bano, one thing you have to understand is maybe you are able to clone your genes into your expression vector. And whether the protein can be functional, there's another story. So how do you ensure that your single domain VH antibody of this TCR light can be stable after expression? The most important is to maintain the functional.
Actually, I do not know if I answer like this. It
doesn't matter. You just
reach
do
your
whatever.
yeah.
So today,
My
today we are not.
understanding, my understanding for this is because during biopanning they have already used bacterial expression system to amplify the phage, and then since we use a bacterial expression system, these antigens are also these antibodies are displayed on the surface of this phage, and then so this suggests that, and then during biopanning we these antibodies are able to bind to their antigens. So these antibodies should have no problem on their functionality lah. And through my understanding, I'm not sure about what are the next step of trying to check what is the stability and all that. Yeah, but to my understanding, the functionality shouldn't be a problem at this stage.
Okay, Ian. Okay, the last question for you is like, now can you go to the objective one?
Objective one.
Because this part you've done already, right?
Okay, this one.
Okay, the vector that you use for the expression is pcDNA 3.1, right?
No, it's the green color one, PET51B, because previously these genes were inserted to a pcDNA 3.1, which is a mammalian expression vector. So actually now I'm just cutting it out and paste it into the bacterial expression vector.
Okay, now you're cutting out the whole gene, and this gene is a VH only genes.
Oh, VH only.
Okay, VH only genes. So before that, your seniors using the VH only genes to express in what system? Mammalian.
Which study?
I don't know
study
because you are using the same gene, right? Do you modify the genes to be a shorter?
No.
Okay.
I just use the because the whole strand of the genes inside the pcDNA actually includes the Fc region as well.
FC reason.
So yeah, I removed the Fc and just produce the VH region.
I see.
Mm.
All right. So you already do the cloning, and I heard that you already got the result of the sequence. So the next step is what kind to transform into what?
And E. coli.
E. coli. Okay. Have you already do the transformation?
Yes.
So how does it going?
It's okay. It's alive.
It's alive. So have you determined the vector inside the bacteria?
What do you mean by determine the vector inside the
Whether
bacteria?
the transformation is successful or not?
Oh, yes. I mean, we use culture agars with ampicillin supplemented culture agars. Then those E. coli that have this vector inside of their, I mean, the successful transformation should have survived on the agar ampicillin resistant. They should be ampicillin resistant and able to survive on this ampicillin supplemented agar. So I think it should be a successful cloning. I mean a successful transformation in this stage.
Okay. So thank you so much, Ian, and I'm done. And thank you, Dr. Koo.
Thank you, Ian. Any evaluator has additional questions? Ian, do you have
I don't have.
co-supervisor?
Oh,
Yeah. Dr. Venu has questions.
No. I said no, I don't have. Thank you, Dr.
You don't have. Okay. All right. So maybe with that we thank Ian.
Okay. So
Thank you.
I'll stop my
Yeah,
presentation.
right.
Okay,
So we go to the next speaker. Right. Dr. Tai, right? Are you there? Dr. Liao.
Hi, thank you, Dr. Koo. I won't be able to chair the next one because I'm currently at the airport and I'll be flying off.
Yeah, no
So,
problem.
thank you very
Dr.
much to
Liao
everybody.
can chair the sessions.
Okay. Thank you, Dr. Liao.
No worries,
Okay.
Dr. Have a good trip. All right. And
Thank you.
so who is the next speaker? Is Gao Zeyu, right?
Yes. Hello, doctor.
Yeah, can you show your slide, please?
Oh, wait for me.
Can you share your slides?
I know. Okay.
Okay, without further ado, and this session will be done by our candidate Gao Zeyu on the title of the growth of inhibitory and drug sensitivity effect of quercetin on cancer cells line. And this project is supervised by Associate Professor Dr. Koo Yin. So without further ado, I will pass the floor to Zeyu, and here's yours, and then you can start your presentation. Thank you.
Okay, thank you, doctor. Thank you all doctors or professors very much for attending my speech. I'm also and my supervisor is Dr. Hu. Today my speech title is the growth inhibitory and drug sensitivity effects of quercetin on cancer cell lines. Why am I doing this research? Because the cancer problem in today's world has become increasingly serious. The World Health Organization says there will be estimated 20 million new cases of cancer and 9.7 million deaths in 2022. Globally, about 1 in 5 people will develop cancer in their lifetime. About 1 in 9 men and 1 in 12 women will die from cancer. Cancer has become the second leading cause of death worldwide, especially in economically underdeveloped areas. There are two figures show the incidence and mortalities of cancer worldwide and their relationship with gender. Also because of the quercetin. Quercetin is a plant flavonoid belonging to the flavonoid group of polyphenol and has strong anti-cancer properties. It also can effectively inhibit the growth and proliferation of viral cancers. Quercetin can also reduce cell viability, autophagy, or promote apoptosis by regulating signaling pathways. Next, we need to have some understanding of sensitivity and gene expression on immune bus. Sensitivity is an in virtual drug discovery platform representing 9 tumor types, which is used to continuously screen potential adjuvant drugs and study drug responses. The gene expression on bus stores the genetic data of the target cell lines we need. About my problem statements, I found a large number of epidemiological studies highlighting the potential of quercetin to reduce cancer cell viability, growth, and proliferation. However, only a few cancer were focused on. More human cancer cell lines that might respond positively to quercetin treatment have yet to be elucidated. As such, our preliminary study aims to investigate the effects of quercetin on 60 different human tumor cell lines using the information in the NCI-60 database to identify and characterize different cells growth inhibitory effects upon quercetin treatment. Specifically, regarding my research progress, I have some question. Does the NCI-60 panel of cancer cell lines provide enough information for the selection of best cell line upon quercetin treatment? What is the gene expression of the selected cancer cell line upon treatment? What are the levels of the growth inhibitory effect of quercetin-rich subfraction of red-impure ethanolic crude extract on the selected cancer cell line compared to pure quercetin? What are the levels of the those gene expression in extract on the selected cell line compared to pure quercetin? For my hypothesis, this study hypothesized the new approach to identify the cell line from the NCI-60 panel of cancer cell line that displays the best effects upon quercetin treatment. The gene expression profiles associated with the growth inhibitory effects of quercetin on the cell line could then be analyzed based on the gene expression on immune repository. Compared with the pure constant, the growth inhibitory effects and some gene expression of the extract can also provide a fresh perspective cancer research. About the research aiming in the objects. My research aiming to comprehensive identify the growth inhibitory effects and gene expression of constant. In cell line from the sensitivity panel, the gene expression on mass database, cell-based assay, and PCR. Specifically, the objectives to there are to identify the cancer cell line that shows the best constant growth inhibitory effect in NCS60 panel, and to analyze the gene expression profile of select cell lines. And next is to investigate the growth inhibitory effect and field gene expression of extract compared to pure constant. This is my flowchart. And here is my research methodology. First step is cell treatment. 5 milliliter of EM medium supplemented with 10% fetal FBS, 100 units per milliliter penicillin, and 100 microgram per milliliter troptomycin will be used to maintain the cell line. The culture will be incubated at 37 cell size in a humidified atmosphere of 5% CR incubator until the cell growth research approximately 85 to 90% confluence. Second step is cell viability test by MTTSE. The cell will be seated in 96 well plates at a density of 10,000 cells per well. Then adhere overnight. The culture will be incubated with 2 milliliter of antibiotic-free media and only adding FBS and subjects for treatments. When the cell reach 60 to 80% confluence, change the media. The cells will then be treated with gradient solutions of extract and pure constant. The cell viability for 1, 2, and days will be accessed by MTTC. After that, the solution incubating at 37 Celsius for 2 to 4 hours. The liquid in the wells removing, and then the MSO will be added to each well to dissolve the formatting crystal. Using microplate reader to measure the absorbance at 570 nanometer. Finally, using the nonlinear regression plot, the dose response curves are constant, calculating the survival percentage and 40% cell inhibition rates of the two types of treated cells based on the curves. The third step is reverse transcription PCR. Setting in 6 well plates at a density of 100,000 cells per well, and overnight incubating the culture with 2 milliliter of antibiotic-free media, and supplementing with FBS only and subjects for treatments. And then change the media in each well when the cell reach 60 to 80 confluence. The cell will then be treated with extract and pure quantity for 2 days. Then harvesting them for totally RNA extraction using a chiral reagent. At least amplify the Ki67, PCN, and BGP genes in a thermocycler. The reaction is denaturation, annealing, and extension. The PCR products will. Oh, sorry. What the The PCR products will be viralized via gene electrophoresis. Oh. About the expected outcomes. The project is expected to select several cancer cell lines that are not typically used in provide studies. The experiments will also demonstrate that the extracts have a stronger inhibitory effect on the growth and proliferation of the selected cell lines than pure quantity. Within 72 to 96 hours, both compounds will be shown to reduce the expression of several genes associated with cell proliferation and drug resistance. I see my Gantt chart. This is reference. Thank you for teachers for my listening.
Okay, thank you, Yi, for your presentation. Now, shall I open this to the floor and who will be the first examiner? Dr. Koo, no, Dr. Koo is a supervisor. Dr. Chiu or Dr. Shaida? Anyone want to be the first?
Maybe I can start first.
Yeah,
Dr.
Dr. Chiu, please.
Okay. Zoe, thank you for your presentation. Sorry, me too have my throat has some problem, like Dr. Liao. Okay. First of all, quercetin treatment, right? Your methodology. Sorry. So in your intro first, the NCI 60, right? In your introduction, you mentioned that like the panel of NCI 60, you have 9 tumor types. And in another slide, you said there are 60 different tumor cell lines, right? In your idea slide.
Yeah, it's 60 different
60,
types.
yeah, that's why it's NCI 60, right? Then in your intro also you mentioned there are 9 tumor types. So how is this 9 tumor types and 60 different tumor cell lines is segregated in the panel? Can you explain a bit about NCI 60, about the 9 tumor types and 60 different tumor cell lines? Right, on NCI 60, you said there are 9 tumor types, right? In this slide.
Mm,
Any idea generally what are the 9 tumor types?
I asked the six cell lines comprised of the 9 tumor type.
Yeah, any idea, rough idea? What are the tumor types in this NCI 60 that you are going to use?
Because I haven't start my experiments, so I'm not very
That for the NCI 60, there's only 9 tumor types, right? Because in your objective and in your expected outcome, or you expect to do other cancer cell lines. I mean, cancer cell lines that are, different from the commonly used one, right? Like you mentioned colon, breast cancer, prostate cancer, lung cancer. You want something, you expect to find some other cancer types, right? Within NCI 60.
like the I remember the monomers and I think I can choose the about 2 to 3 the cell lines. It's have the better sensitive than other cell lines.
Okay. Okay. Then maybe we go to the objective, main objective. Can you go to slide number 5? Slide number 5, right? Your main objective. This is slide number 5 because Your main objective. Sorry, it's not number 5, right? Yeah. Your main objective. It doesn't sound clear here because to comprehensively identify the growth inhibitory effects and gene expression of quercetin. I think it's not of quercetin, right? It's the treatment with quercetin.
Yeah, upon the treatment.
Yeah. I think something's missing is treatment with quercetin, not off quercetin. That's it. Yeah. So it doesn't sound accurate here. I think you have to modify a bit.
Yeah.
Then for your objective number 2, right? You try to identify the gene expression based on the GO, right? Repository.
Yeah.
But in objective number 3, you actually have identified Ki67, PCNA, PGP, right?
Yeah.
So for objective number 2, actually is unknown to you, right? What kind of gene will show, can you screen from the GO profile, right?
Actually, we must choose the best size from the SS60, and yeah, there have been choose, and we use these satellites to we use these satellites and find their normal gene expression in the gene expression omnibus.
Yeah, but it looked like you also have already identified like Ki67, PCNA, PGP genes to monitor the expression level, right? But
they just sorry.
pardon?
They just example, yeah. I don't know. These is whether or are my 就是the the gene expression we need, yeah.
Okay. And then we go to slide number 8. Because I cannot see the number of the slide clearly, I would say maybe number 8. You go to slide number 8.
Which eight?
Number 8 of methodology. No, not number 8. I just would like to know what are the concentration of pure quercetin that you are going to test, because you said that you are going to use 10, 20, 40, 80 micromolar of onion extract, right? And you want to compare them with pure quercetin, right?
Yeah, compared to quercetin.
Yeah. What concentration of pure quercetin that you're going to use for the comparison? For the onion extract, you mentioned that you're going to use 10, 20, 40, 80 micromolar.
You mean?
Yeah,
you mean
I
this
think,
one?
yeah, this one, yes. This is for your onion extract, right? So how about the pure quercetin? What concentration of pure quercetin?
I think the same.
Yeah, are they the same? You're going to use 10, 20, 40, 80 of pure quercetin as well?
Yeah.
Yeah, then how do you prepare the onion extract? Are you going to prepare the onion extract?
I think my supervisor will from the my supervisor's previous study, I think she will give me.
Pardon?
I think my supervisor will give me the
Will
for,
give
for,
you
for
the
my
onion.
spider. Yeah.
Okay, maybe you have to find out about the onion extract preparation, whether you are going to freshly prepare the onion extract or whether the onion extract have been purified or what, because you are going to compare the pure quercetin, right? Because this info is not in your presentation. That's why I think you have to find out, right, about the onion extract. Okay, has to understand more.
Yeah.
Then and then expected outcome, right? Your aim is to find other cell lines. You hope to find other cell lines other than the usually used one, like cancer cell, breast cancer cell, colon cancer, right? So what other cell lines that you expect to screen out from your panel? Other than the usually used cell lines. You want to find, right, the first objective, what other cancer cell line that you expect, or you hypothesize that you can select other than this usually used one? Any idea?
I think everyone the drug sensitivity better than the Euro cell lines.
May I know, like all these prostate, cervical, lung, breast, and colon cancers, they are not in NCI 60 panel, right? Are they inside the NCI 60?
Oh yeah, they are inside.
They are inside the NCI 60. So how if you find out that actually the most affected one is also among these few? So what will happen then?
You mean how I found the best
Yeah, because
cell
you
line?
want to find something else, right?
Yeah.
So if you screen, then you find out actually the best one is breast cancer. So how? Do you proceed with breast cancer or how?
Mm, this is my this was
The same questions go to the gene expression. If like, let's say after you screen, right, with the GO profile, then you find out actually the best the gene expression is not KI 67, PCNA, PGP,
yeah.
then what's next?
I actually don't know what. Yeah. If I had choose the best cell line and their yeah and find their expression gene expression the the gene we need why uh, why we must use the like the genes KI 69 67 they're just the example.
Okay, so my last questions will be the last expected outcome, right? You want to prove that gene expression associated with cell proliferation and drug resistance. Can you give example? What are the genes for cell proliferation and genes for drug resistance? The third expected outcome, right? A few gene expression associated with cell proliferation and drug resistance. So maybe can tell us one example of cell proliferation gene, and one example for drug resistant gene that you find out from your your literature review?
The proliferation is yeah. This is why this is why I give you the example. The proliferation is the Ki 6F7 and the P7. This is the about genes proliferation, and the PGP is the about the drug resistance.
Oh, okay, okay. Yeah. Thank you, Zeri, for your explanation.
Thank you.
Dr. Liao, I have no further questions. Thank you.
Thank you so much, Dr. Chiu. And so shall we invite the second presenter, Dr. Shida, right?
Yeah.
Not presenter, examiner. Thank you.
Yes. All right. Thank you, Dr. Liao. So hi, Gao. I have just a few questions because most of my questions have been asked by Dr. Chiu, but it's the same thing. I can't really see. What I understand is that from your NCI 60, you're trying to identify which cell lines, and then from the GEO, you're trying to see the gene expression profile. So in this, on your second objective, what is your aim? What is the correlation with the reverse transcription that you are focusing on Ki67, PCMA? Can you explain on your second objective?
Yeah. Let me explain my methodology.
Okay.
So this is actually the methodology, and sorry, wait for me. First, I know we know the gene expression on mass is a repository, and but no, this SQT have many databases in the about with the cancer cell lines, and we use them to identify the best the most sensitivity and the drug sensitivity and the most inhibitory the cancer lines. Yeah, this is the first step, and second is MTTSA. We do MTTSA, and we will get three group results. One is the control group, two is the extract group, and secondly, third is the pure consisting group. Then we use the then we can get their OD value, and we use this observance to plot their this curve, and we can use this curve find the cell viability 50% this area, and we found their this IC50 value. And the last step is we use the IC50 value to treat the to treat the cell line, to treat the cell line. Yeah, we choose the cell line. To treat it, we choose the cell line and go through the MTTC. We get the viability. The viability show the cell inhibitory effects, and after we use the concentration or concentration with the IC50 value. We use this concentration to treat the cell lines. We can get the we can use the RT-PCR to get the gene expression changed. So final we use the gene expression changed and compared to the gene expression normal. Yeah, the normal is we found from the gene expression on bus. Yeah, then we can find which gene is which gene was changed, and we can next we can study which gene is which gene is have the relationship or with the drug sensitivity and or the inhibitory. Yeah.
Okay, so you mean these three genes refer to the inhibitory and proliferation marker? Is it?
Sorry, I don't know. Clear.
It's okay. Okay, then so what about the statistical analysis? What do you think is a suitable statistical analysis that you can apply in your study? Because in your proposal, written proposal, you put statistical analysis, but you didn't specifically mention what kind of test that you plan to do.
That's because actually because I'm not having start so the
So it's
I'm not
specific.
very clear about the that this is
Okay. I understand.
statistics. Yeah, I'm not very clear about this, so I don't show this.
Okay, but it's something that you need to consider and maybe put in your when you have like two different groups, treated and untreated, then you only need to choose statistical analysis and compare between two groups. But that one you can discuss with your, And then obviously I noticed your slide number is very not in order. I think that why you need to correct before you send your final slide to your supervisor. The slide number is not accordingly, it's not in order.
Yeah.
And your methodology, I guess, can you go to the previous slide on your first slide on methodology?
First?
Methodology, the cell culture. Okay, I guess this is for your objective number 2, right? What about the first objective on NCI 60? Can you explain how we're going to do that? To search for the potential cell lines? it's basically a bioinformatic, right? But can you just briefly explain how you're going to do the bioinformatic analysis?
In the NCSD database, they have the they also have the cell relative 50%. Yeah, I say 50 value. Yeah, I think I search the cancer cell lines by the I say 50 value.
Okay. And my last question on your expected outcome. When you mention both compounds, does it mean the subtraction and the pure quercetin, right? The last point, the third point, the both compounds will prove the reduction.
Which?
The third point, subsequently, both compounds will prove the reduction. What you mean by both compounds?
Yeah, this postpone is activate and pure quercetin.
Pure. All right. Okay. Thank you for your answer. I think that's all my question, Dr. Liao.
Okay, thank you, Doctor.
Dr. Liao, back to you. It's okay, maybe Dr. Liao is not here. Yeah, I think that's all questions from me and from anything from Dr. Xu? No, right?
No more.
All right,
Yeah.
then. Yeah, that's the end, I think. Thank you, Gal, for your presentation, and I wish you all the best for your project. And I think that's all. Thank you.
Thank you, Dr. Shahida and Dr. Chiu for the
Thank you, Dr. Gu.
comments.
Yes.
Thank you, Doctor.
I think we can just end this session.
Mm.
Thank you, everyone, for attending. And then, Gal, you can close your slide and
we can,
yeah.
we can close.
Later, Bob maybe can send the recording to me, and then I will let the student listen to his talk and also how he answered the questions again.
Okay.